Have you ever marveled at the complexity of a butterfly's wing? The delicate patterns and vibrant colors are a testament to nature's artistry. Or the intricate workings of the human brain? This organ, with its billions of neurons and synapses, is the control center of our thoughts, emotions, and actions. It's astounding to think that these complex structures arise from a single fertilized egg cell. This tiny cell holds all the information needed to create a complete organism. This incredible journey from a single cell to a fully formed organism is what we call embryonic development. It's a process that involves countless steps and precise coordination. Understanding this process is like unlocking the secrets of life itself. Scientists dedicate their lives to studying these mechanisms, hoping to uncover the mysteries of our existence. We often take for granted the miracle of development. The birth of a baby or the hatching of an egg is a culmination of a complex series of events. A tiny seed transforms into a towering tree. This transformation is a marvel of nature, showcasing the power of growth and development. A frog egg becomes a leaping amphibian. The metamorphosis from egg to tadpole to frog is a dramatic and fascinating process. These transformations are anything but simple. They require precise timing and coordination at every stage. They involve a tightly orchestrated series of events, guided by an intricate network of molecular interactions. Each step is crucial for the proper development of the organism. At the heart of this process lies our DNA, the blueprint of life. This molecule contains all the instructions needed to build and maintain an organism. But it's not just about the genes themselves. The regulation of these genes is equally important. It's about how those genes are regulated, turned on and off at precise times and in specific cells to create the incredible diversity of cell types and structures in our bodies. This regulation ensures that each cell knows its role and functions correctly. Join me as we delve into the fascinating world of embryonic development, exploring the molecular mechanisms that drive this incredible transformation. We'll look at the latest research and discoveries in this field. We'll uncover the roles of key players like transcription factors, signaling pathways, and epigenetic modifications, all of which are essential for proper development. All these elements work in concert to shape a single cell into a complex living being. This journey from simplicity to complexity is one of the most remarkable phenomena in biology. The journey begins with fertilization, the fusion of sperm and egg. This momentous event marks the beginning of a new life, a process that is both intricate and awe-inspiring. This union creates a single cell, the zygote, containing the complete genetic blueprint for a new individual. Within this tiny cell lies all the information needed to develop into a complex organism. This newly formed zygote doesn't waste any time. It embarks on a rapid series of cell divisions known as cleavage. Each division is a step towards complexity, transforming the single cell into a multicellular structure. During cleavage, the single-celled zygote divides repeatedly, forming a cluster of smaller cells called blastomeres. These blastomeres are the building blocks of the embryo, each one carrying the potential to become a part of the new organism. Think of it like inflating a balloon. You're not adding more material, just redistributing what's already there. The zygote's initial size remains the same, but the number of cells increases exponentially. Similarly, the zygote's cytoplasm is divided among the blastomeres, creating smaller but more numerous cells. This redistribution is crucial for the next stages of development, ensuring that each cell has the resources it needs. Cleavage leads to the formation of a blastula, a hollow ball of cells surrounding a fluid-filled cavity. This structure is a significant milestone, marking the transition from a simple cluster of cells to a more organized form. This seemingly simple structure sets the stage for the next crucial stage, gastrulation. Gastrulation is a transformative process that will lay the groundwork for the embryo's body plan. During gastrulation, cells within the blastula rearrange themselves into three distinct layers: the ectoderm, mesoderm, and endoderm. This reorganization is essential for the proper development of the embryo's tissues and organs. These germ layers are the precursors to all tissues and organs in the developing embryo. Each layer has a specific destiny, contributing to the formation of different parts of the body. Think of these layers like the foundation, framing, and walls of a house. Just as a house needs a solid foundation and structure, the embryo needs these layers to develop properly. The ectoderm, the outermost layer, will give rise to the skin, nervous system, and sensory organs. This layer is responsible for forming the body's protective covering and its communication network. The mesoderm, sandwiched in between, will form the muscles, bones, circulatory system, and reproductive organs. It provides the structural and functional support needed for movement and circulation. The endoderm, the innermost layer, will develop into the lining of the digestive and respiratory tracts, as well as associated organs like the liver and pancreas. This layer is crucial for the body's nutrient absorption and respiratory functions. Each step in this process is meticulously coordinated, ensuring that the embryo develops correctly. From a single cell to a complex organism, embryonic development is a testament to the wonders of biology. As the embryo continues to grow and differentiate, it will eventually take on a recognizable form with a beating heart and developing organs. This journey from a single cell to a fully formed individual is nothing short of miraculous. With the basic body plan established, the real magic begins. Organogenesis, the formation of organs. This intricate process relies heavily on the instructions encoded within our genes. Those segments of DNA that carry the blueprints for building and maintaining an organism. But it's not as simple as genes being switched on or off. Imagine a vast library filled with cookbooks. Each cookbook represents a gene, and each recipe within represents a specific protein that the gene encodes. Just like you wouldn't use every recipe in every cookbook, not all genes are active in every cell at all times. This selective gene expression is crucial for proper development. During development, different sets of genes are activated or silenced in different cells, depending on their location and the stage of development. This precise regulation ensures that cells differentiate, meaning they specialize and acquire the unique characteristics and functions that define them as muscle cells, nerve cells, skin cells, and so on. This intricate dance of gene activation and repression is orchestrated by a complex network of regulatory mechanisms. These mechanisms control which genes are transcribed into RNA and ultimately translated into proteins, the workhorses of the cell. Let's meet some of the key players in this molecular orchestra. Section 4. Master architects, transcription factors, and cell fate. One of the most important groups of regulators in cellular biology are transcription factors. These are specialized proteins that bind to specific DNA sequences and control the rate of gene transcription, which is the first step in gene expression. Think of them as master architects, interpreting the blueprints encoded in our DNA and directing the construction crew, which in this case are the various molecular machines within the cell. Transcription factors can either activate gene expression, turning up the volume of protein production, or repress it, effectively silencing the gene. This regulation is crucial for maintaining cellular function and identity. The activity of transcription factors is often influenced by signals from neighboring cells or the external environment. These signals can include hormones, nutrients, and other molecules that convey information about the cell's surroundings. This dynamic interaction allows cells to respond to their surroundings and adjust their developmental program accordingly. For instance, a cell might change its function or even its identity in response to new signals. A fascinating example of this is during limb development. A specific signaling molecule called Sonic Hedgehog, yes, it's named after the video game character, plays a critical role in determining the identity of fingers and toes along the hand and foot. This molecule acts as a signal that guides the activity of transcription factors in developing cells. Different combinations of transcription factors can activate unique sets of genes, leading to the formation of distinct cell types. This is how a single fertilized egg can give rise to the myriad of cell types found in a complex organism. It's like using the same set of building blocks to create a house, a skyscraper, or a bridge. The diversity of structures comes from the different ways the blocks are arranged and assembled. The arrangement of those blocks, guided by the architect's plan, determines the final structure. Similarly, the specific combination and activity of transcription factors determine the identity and function of a cell. Mutations in transcription factor genes can have profound effects on development, often leading to birth defects or disease. These mutations can disrupt the normal regulation of gene expression, causing cells to behave abnormally. Understanding how these master regulators function is crucial for deciphering the complexities of development and disease. By studying transcription factors, scientists hope to uncover new ways to treat genetic disorders and improve regenerative medicine. Section 5. Cellular communication signaling pathways in embryonic development. But transcription factors don't work in isolation. They are part of a larger communication network within and between cells known as signaling pathways. These pathways involve a cascade of molecular events like a game of molecular tag, where one molecule activates another which activates another, and so on. These signaling cascades relay information from the cell surface to the nucleus, where they can influence gene expression. Imagine a relay race where the baton carries a message from one runner to the next. Similarly, signaling molecules carry messages that instruct cells to grow, divide, differentiate, or even die. One well-studied signaling pathway is the WNT pathway, involved in a wide range of developmental processes, from establishing the body axis to controlling cell fate decisions. Disruptions in WNT signaling have been implicated in various cancers, highlighting the importance of these pathways in both development and disease. Another important pathway is the Notch pathway, which plays a crucial role in cell fate determination, particularly in the nervous system. Imagine two neighboring cells, one destined to become a neuron, and the other a supporting glial cell. Notch signaling helps to solidify these fates, ensuring the proper balance of cell types in the developing brain.